Hello. The chemical nature of nucleic acids happens to be the fundamental elements or the molecular components that make up the subunits. When we say subunits here, we mean the nucleotides. The nucleotides are made up of three distinct components, or simply put, we can call them as they are nucleosides plus phosphate group. And nucleosides themselves are made up of two further components. They are the nitrogenous bases and pentose sugar. So basically, if we talk in that ways, we have three distinct components, and they are these three. We have to talk about the nitrogenous bases, the sugar, and the phosphate group that make up the subunits or the components, basic components of nucleic acids. And in that basic components, we also have some more of these. Further classification for nitrogenous bases, we have purines and pyrimidines. So the purines have a double ring structure as shown in here, and they are adenine and guanine, whereas pyrimidines have a single ring structure, that is, they are cytosine, thymine, and uracil. Adenine and guanine are present both in DNA as well as RNA. So let's write it here. Say, for example, this is DNA and here is RNA. In terms of bases, what is the chemical difference between DNA and RNA? The difference is that in DNA we have thymine and but not uracil, and in RNA we have uracil but not thymine. And the chemical difference between thymine and uracil is this one methyl group. So we can say it as the difference in a way is that there is one additional methyl group in DNA as compared to RNA in terms of nitrogenous bases alone. And if we look at the sugars, the pentose, we have differences in DNA and RNA. That is, basically it's about in DNA we have deoxyribose and in RNA we have ribose. And the difference between these two is that in RNA you have one oxygen more than the deoxy. So that is the only at the level of chemicals components, if you are speaking strictly, then that is how we can say the differences are. And we also have to keep in mind the numbering, and previous videos would help in that. Some of the numbers we should remember of that of ribose are very important. The 2' carbon, which is where the oxy or deoxy, the ribose or deoxyribose is determined by that. And 3' OH, or the third carbon, 3' carbon, is important because polymerization would happen at this OH. And 5th carbon is also involved, is important because the 5' phosphate that is present and involved in polymerization. So basically the 3, 4, and 5 carbons actually form the base, form the phosphate sugar backbone of nucleic acids. We will see them further. I am Dr. PCMR, Assistant Professor at Shastra. The first things we will discuss in today's class are that how does polymerization occur? What we have seen so far is that we know that we understood the chemistry of the subunits, the monomers or the nucleotides, and how do they polymerize. There are two important bonds that we need to understand, and in the first with the basic components here, we need to understand the bonds that are, this is the glycosidic bond. And we will also know a few more bonds that are important. One is the phosphodiester bond and then the hydrogen bonds. First, we will see about the phosphodiester bond because that is what will allow the formation of a polynucleotide. Here we see a base, and this is guanine, and this one is guanine 2, and this is a dinucleotide of a guanine, two guanines. Guanine is in monophosphates, and here we have, this is one component, this makes up one base, one nucleotide, and this makes the other nucleotide. So the 5' carbon is here, and which is what will form a phosphodiester bond with the 3' OH of the preceding deoxyribose. I'm calling it deoxyribose because there is no OH indicated here. Yes, so this is a phosphodiester bond, and I hope we recognize that in the phosphate backbone, this is a dinucleotide, and we can have many more nucleotides coming and joining here, and that way we have a chain. So if we look at the phosphate backbone, we will see something like this. Assume each of these is a sugar, this the ribose, and middle we have phosphate group. The sugar is something like this. It should have been a pentose, but just for the sake of understanding, we'll put it this way. What I mean to, or the intention here is, the sugar is part of the backbone, right? And so we'll have something like a phosphate group here that is joining them. We are only at this point we are only depicting the the phosphate backbone of the nucleotide, polynucleotide. And if I were to indicate the bases, nitrogenous bases, they would have been attached to the ribose, something like this. This is just a depiction of how these moieties are arranged in a polynucleotide. This is important to understand. If we remember the definition of any information, it should have subunits and it should have a sequence, and specific sequence will come to later. So we have the subunits, and now we are trying to understand the chemistry of how sequence is formed. So the black ones here indicate the bases, and the red one is indicating the deoxyribose or ribose, as the case may be, and the blue is indicating the phosphate. The nitrogenous bases are not part of the sugar phosphate backbone. Yes, but the bases have something very important, and if we remember again this definition of genetic information, it should be inheritable information. So for that we need to understand the importance of bases, the nitrogenous bases, and the more specifically the importance of the hydrogen bonds they form. As we already know now that G base pairs with C using 3 hydrogen bonds, and A base pairs with T using 2 hydrogen bonds. That means there is a way of determining the sequence of one strand. Say for example, I have a sequence like this, then the opposite base or the complementary strand is determined by what is present here. A, therefore we'll have T, and T therefore we'll have A, G, C, T, and G. So that is one sequence is resulting in or determining the sequence of the others or the complementary strand. That happens because of the way the bases are present in these nitrogenous bases or the side groups. The hydrogen bond formation between the amino group of cytosine and the keto group of the oxygen here, they will determine the hydrogen bonds. So there are three ways of three hydrogen bonds that can form between cytosine and guanine. Similarly, between adenine and thymine, and also in RNA, adenine and uracil can form two hydrogen bonds. And that is how the information that is present in one sequence, one strand, can determine the sequence in the other strand. So if we look at in terms of the information, the phosphate backbone is determining the sequence, is able to show it as a sequence, and the hydrogen bonds that are present are making it inheritable, right? Because the information of one strand is resulting in another strand. Or if we have, like we discussed before, double-stranded DNA can give rise to two double-stranded DNA, and they will be identical to that of the parent DNA. So that way, the inheritance of information is happening because of these hydrogen bonds. The first time these polymerization, these hydrogen bonds were forming, specifically between these bases, I think that is the first seeds of life. And from then on, we have diversified to what we are right now. So it's a beautiful and incredible way of seeing how life must have originated, how from matter, information, and now life as information came into existence. And that all is present in this beautiful molecule of DNA, the only molecule which is energy, which is matter, and which is information, not just information, which is inheritable information. The double-stranded DNA, we will have further discussions on this, but it is important to see how the structure becomes information, how the chemistry results in information. The appreciation of at that level gives us more insights into how, or allow us to ask further more questions. Are we just matter? Do we have any purpose in life, or what else can be done? Something like that. So the phosphate backbone, as we know, it is on the outside, the one that is shown in solid here, and the bases are in the inside. We will discuss about the structure of DNA and the reasoning different scientists used and how they have made it theoretically, and later was proved that it is correct. That is how it is. On the end, I would want to show this picture, a GIF. It's a very nice way of seeing it, the DNA that is spinning, and you can see that the bases are inside, and then we have the cyan or magenta that is showing the phosphodiester backbone. We'll discuss more on the structure of DNA in the later ones. Thank you very much.